It is my great pleasure to introduce Dr. Mia Levine. Mia got her undergrad from the University of Pennsylvania and a master's degree from the University of Illinois. She got her PhD in David Begun's lab at the University of California in Davis, studying genome evolution in Drosophila. From there, she joined Harmit Malik's lab at Fred Hutch for her postdoc, where she continued this focus, but this time looking at heterochromatin evolution in Drosophila. And Mia has since come full circle back to the University of Pennsylvania, where she is an associate professor. Mia has won many awards, not just for her research, but also for her mentorship and teaching. And I won't take too much more time by elaborating on these awards, but I did want to add that I heard Mia and her postdoc, Kara, talk about their work at a conference last year, and I was really excited by their science. And I'm hopeful that everyone here will share that enthusiasm for her work. Welcome, Mia.
Thank you, Parvutha. Thank you for the invitation to participate in this really wonderful seminar series. It's a real privilege to be here and have the opportunity to share some of our work looking at the evolution of genome integrity. So I'll begin talking about intragenomic conflict with the idea that a single genome appears to be a sort of harmonious cooperative of distinct genetic elements. So we think about development, our distinct genes collaborate to build a robust and fertile individual. If we think about evolution, our distinct genes can accumulate adaptive mutations to build an even more robust and even more fertile individual. Of course, the more robust and fertile the individual, the more copies of all of these genes in the next generation. And so in these ways, across both development and evolution, our genes contribute to a communal good. At the same time, it's inarguable that much of our DNA contributes to building neither a robust nor fertile individual. And while much of this DNA is effectively neutral, it's not deleterious enough to be removed efficiently by natural selection. A fraction of this DNA persists in our genome and even amplifies over evolutionary time, despite the fact that it compromises our Darwinian fitness. So these genes or genetic elements are typically called selfish, and they come in a variety of flavors, arguably the most ubiquitous of which are the mobile genetic elements like retrotransposons that copy and paste themselves around the genome. They increase their copy number, often while wreaking havoc on genes or gene regulatory information and even promoting ectopic recombination. So rather more mysterious, but also selfish are the meiotic drivers. These selfish elements bias their transmission from one generation to the next. A classic example of a meiotic driver is a selfish centromere. So a selfish centromere is a element that biases the transmission of the chromosome that it's on. So remember, centromeres are the site of microtubule attachments that pull chromosomes apart during meiosis and mitosis. During meiosis, these microtubules pull the chromosomes either towards the egg, which represents the next generation, or instead towards the polar body, which is effectively an evolutionary dead end. And as Mendel would have predicted, these chromosomes have a 50% chance of moving towards the egg or towards the polar body. However, selfish centromeres can arise. These selfish centromeres are associated with a proliferation of the DNA satellites that make up the centromere. This selfish satellite proliferation is selfish because these chromosomes that have the selfish DNA are moving now towards the egg and away from the polar body at rates of up to 90%. And this selfish centromere can actually confer fertility costs as well. So these very different types of selfish genetic elements share a few features I'd like to highlight. The first is that they are both repetitive DNA. They are non-unique sequence in the genome. they both increase their copy numbers and gain a transmission advantage relative to other single copy elements in the genome. And finally, this advantage can come at a fitness cost. This fitness cost can trigger the host to adapt to suppress the selfish element's proliferation directly, or maybe the collateral damage of the proliferation of that selfish element. And this adaptation on the host to suppress the selfish element can be met by counter adaptations of the selfish element, perpetuating what we like to call molecular arms races. So we can visualize these molecular arms races or intragenomic conflicts in the following way, where at some time point in the past, there's a host protein that is suppressing successfully some selfish repetitive DNA element. Over time, this repetitive element can innovate and escape this suppression. Sometimes this escape can trigger a fitness cost, again, either a direct fitness cost due to the proliferation of these repetitive elements, for example, sort of unmitigated activation of a transposon, or alternatively, the fitness cost can arise from, again, the collateral damage of a repetitive element proliferating. This fitness cost will then select for innovation on the host side, and this may look like adaptive protein evolution, which results in very different looking proteins in yet closely related species. They've accumulated many amino acid changing mutations, and this can lead to re-engagement and suppression of the repeat once again. So these intragenomic conflicts raise many questions and goals for my lab. We're really interested in understanding the sort of experimental evidence of this intragenomic conflict model. It's a very compelling cartoon, but there's been minimal kind of experimental investigation of the reality of this model. And maybe by studying it experimentally, we can offer some elaborations. Secondly, in the process of doing this, we can identify the who in these conflicts, who are the genome components that are engaged. And finally, we are interested in the mechanisms by which the host can restrict these selfish elements and ultimately mitigate their collateral damage. And again, in the process of addressing these, we hope to reveal the biology that's been sculpted by this intragenomic conflict that plays out over evolutionary time. Historically, however, these intragenomic conflicts have been quite challenging to study, in part because when we go out to a natural population and collect a sort of healthy, fertile individual, they're typically in this suppressed state. So these conflicts can be quite cryptic when we attempt to, find them in a contemporary genome. So to get around this crypsis, my lab uses the DNA signature that's left behind by these intragenomic conflicts. So this might look like adaptive protein evolution on the host side, or the non-random accumulation of these amino acid changing mutations, as well as really striking divergence on the DNA repeat side between closely related species. We then go in and actually swap the native version of one of our candidate genes with one from a close relative that's highly diverged. We call this an evolutionary mismatch between the host protein of one species that is sort of naive to the landscape of repeats of another. And we hope that by making this evolutionary mismatch, we're going to resurrect that transient state of proliferation or deleterious byproducts of proliferation in order to study them in real time. For these studies, we primarily used Drosophila melanogaster, which has abundant genomic resources for doing our population genetics and molecular evolution to identify those signatures of divergence, as well as many genetic tools for manipulating genomes. So we've taken this approach using multiple systems, and we've previously published on some work where we've discovered evidence of host adaptation to directly suppress selfish element proliferation. And in this case, this was for the transposons that live at the ends of Drosophila chromosomes at their telomeres. In today's talk, I'll walk you through instead some of our work looking at what the deleterious incidental byproducts are of once a DNA satellite has expanded, what sort of problems does that incur on the host and how the host adapts to mitigate them. So how do we go about discovering a list of candidate host proteins and DNA repeats that we can then study and manipulate? So we honed in on a DNA satellite from Drosophila melanogaster, which has been reported to span 11 megabases of the X chromosome near the centromere. This is called the 359 base pair satellite. And remarkably, this massive array in the Drosophila melanogaster genome is not present in its very closest relative, Drosophila simulans, or in an outgroup, Drosophila erecta, suggesting that this repeat has proliferated specifically along the melanogaster lineage. On the host side, we identified a protein from the literature called maternal haploid. So Yi-King Rong in 2017 published some work on this maternal haploid gene, where he reported that in embryos, by conducting immunofish, discovered really striking co-localization of the maternal haploid protein and this 359 base pair repeat. So we wondered if maybe this association and the high divergence of 359 suggests that these two proteins, this protein in this DNA satellite, might antagonistically co-evolve. So that hypothesis is predicated on the idea that MH actually evolves adaptively itself. So we addressed that question using a classic population genetic test called a McDonald-Kreitman test, which compares the polymorphisms and differences or fixations between melanogaster and simulans, and compares the changes that are silent or synonymous to those that are non-synonymous or amino acid changing. And we find an excess of non-synonymous differences between these very closely related species. This suggests a history of adaptive protein evolution of the maternal haploid gene. So to develop a hypothesis for why they might be co-evolving, we had to understand what MH does. And it turns out that MH is expressed highly in the ovaries of Drosophila melanogaster females, and this protein is deposited into the embryo, where it's really important for the paternal chromosomes, that's the sperm deposits, to participate in mitosis. So in a wild-type embryo that is laid by a wild-type mother, she deposits her maternal chromosomes, as well as a whole bunch of that MH maternally deposited protein. Dad deposits through the sperm his paternal chromosomes, and this paternal chromosome set is actually totally unprepared to do mitosis. It's highly condensed in that sperm head. Many maternal proteins act on this paternal DNA to prepare it to undergo mitosis along with the maternal chromosomes and create a diploid embryo. When mom is mutant for MH, she deposits those maternal chromosomes and dad deposits those paternal chromosomes, but there's no MH protein there. And so dad's DNA fails to undergo proper chromosome segregation, and 80% of these embryos die. However, 20% of them are able to ignore dad's DNA and cycle only the maternal haploid complement of the DNA and ultimately die before hatching. So this functional precedent suggested to us that maybe the melanogaster specific 359 base pair array that's only in melanogaster depends on the melanogaster specific version of this maternal haploid protein in order to segregate properly at this very first mitosis. So to test this hypothesis, we generated that evolutionary mismatch, such that we have a control fly and an experimental fly. In our control fly, we use CRISPR-Cas9 to remove the native gene and replace it with the melanogaster-specific coding sequence, tagged with flag and codon optimized for melanogaster. Our experimental fly is exactly the same, except that it has the simulins coding sequence in an otherwise melanogaster background. This is our experimental fly, and it's called MHSim for the purposes of this talk, and it will always be shown in yellow, whereas the control will be MHMel in blue. So is the first embryonic mitosis disrupted when you have MHSim? So we collected early embryos from mothers that were either mutant for MH or MHMel or MHSim. And you can see on the y-axis the number of embryos and the cycle number along the x-axis. When we have mutant mothers, as the literature suggested, we see an excess of embryos that are stuck at this very first cycle. In contrast, when our control females lay eggs, they have embryos that undergo mitosis normally, creating a distribution that approaches normal. And surprisingly to us, the MHSim transgenic flies also produced exactly the same distribution, suggesting to us that MHSim completely rescues the mutant phenotype. So then, is there any functional consequence of all of this evolution at all of maternal haploid? Well, knowing that maternal haploid is expressed in the ovary, we looked at female fertility, and we found that the MHSim genotype, which you can see on the x-axis, produces fewer offspring than the MHMel genotype, suggesting that there is some sort of defect, but clearly not the first mitotic division. So we looked at the ovaries of these MHSim females that lay those eggs. And here's just a cartoon of a Drosophila ovary where you can see the mature egg highlighted in gray. So when we look at the MHMel and MHSim ovaries, what we can find is a sort of atrophied MHSim ovary. And to quantify this, we just counted the number of mature eggs in each and found a significant loss of mature eggs when you have the wrong species version of MH. But this was really surprising. So we rescued the mutant phenotype, but we are actually finding a phenotype that had not been reported before. And we indeed confirmed that the mutant ovaries that don't have any of this protein are actually completely fine. So this suggested to us that not only is MHSim not a loss of function allele, but it actually might be toxic. So to address that possibility, we overexpress this protein in the ovary. We use the UAS-Gal4 system in flies. So our genotype is a fly that has the native version of MH. It has a promoter hooked up to a transgene. In this case, it's the control MHMel transgene. And then we put into the same fly a transcription factor driven by an ovary-specific regulatory sequence, which is called Gal4. And then we're able to drive expression specifically in the ovary. I'm showing you here an ovariole, which is sort of an egg production factory that makes up the ovary. And we can compare this fly to the one where we've overexpressed this potentially toxic protein. So the results of this experiment were quite striking. When we look at the total offspring from these females, what we find is that when we overexpress the right species version, there seems to be no defect, while we get complete sterility when you overexpress the wrong species version. And in fact, these ovaries make absolutely no mature eggs. So to understand the sort of cell biological basis of this toxicity, we turn back to our flies that have the native promoter driving either MHMel or MHSim. And we are again looking at those ovarioles, those sort of egg production factories where the anterior end has the germline stem cell. And as you move posterior, you get a more and more mature egg. And what you can see here when we just stain with DAPI is excess of condensed nuclei that are consistent with cell death. And we quantified this excess in the MHSim ovaries. To understand what the basis of that cell death is, we looked for a classic trigger, which is DNA damage. We stain these ovaries for gamma H2AV, which is a marker of double-strand breaks in Drosophila melanogaster. And we find an excess of this DNA damage marker in MHSim ovaries, and you can see the quantification on the right. We also, in some data I don't have time to show you, use a checkpoint mutant to show that the egg production block that we observe is truly mediated through a DNA damage pathway. So these phenotypes of DNA damage and loss of egg production really set us up to ask our primary question, which is, do these two components of the genome co-evolve? Effectively, we have this toxic phenotype. We have this phenotype, this loss of fitness. And now we can ask, is it dependent on the presence of 359, which is what we predict if these two components of the genome are truly incompatible, which is to say they've co-evolved along the melanogaster lineage. So effectively, is this toxicity dependent on 359 or something else in the genome? It really could be anything else. So to test that hypothesis, we took advantage of a strain that actually lacks the 359 array, and we recombined its X chromosome with our MHMEL or MHSIM transgenes. And we ask if this can actually rescue the challenges that these MHSIM females suffer from. And so we stained these ovaries for our DNA damage marker and find that actually removing this repeat completely rescues the germline genome integrity as seen through this marker. And here's the quantification of that to the right. We also found that these females produce similar numbers of mature eggs, as well as restore the fertility completely of these otherwise sort of toxicity-bearing MHSIM females. So these data suggest that by introducing a sort of naive version of MH into the melanogaster genome, we generate this subfertility. However, if we effectively make the melanogaster X chromosome simulins-like by removing this melanogaster-specific repeat, we're able to restore fertility. So while these data are certainly consistent with sort of a classic model of antagonistic coevolution between repeats and host proteins, we think this provides a few elaborations. So the first, I'd like to explain by kind of leaning on our intuition, which is that if we have a satellite repeat that's changing and expanding, we might expect that the host protein has to evolve to recognize and keep up and interact with this novel sort of foreign repeat. And so when we swap in the wrong species version, we should see a loss of function. But we didn't see a loss of function. Instead, what we saw was toxicity. So this suggests instead that our naive version doesn't fail to do something, but instead actually interferes with the processing of this repeat. So we want to understand, what might be the basis of this interference. But to sort of have this model go down the road, we have to include the idea that the MHMEL version actually does not interfere or effectively avoids this repeat in the ovary. So to dig into this, we really need to understand something more mechanistic about this toxicity. And to do this, we really had to turn to humans where the MH in humans, which is called Spartan, is much better characterized. And what we learned from these studies of Spartan in mammals is that this is a protein that is important for DNA repair, and specifically is a pathway that's specialized for when proteins become cross-linked to the DNA. So many proteins physically interact with our DNA, become reversibly cross-linked, do some important DNA transaction, and then get removed. But sometimes these proteins actually undergo this irreversible DNA protein crosslink, and there's a specialized pathway that actually chews up these DNA protein crosslinks. So Spartan is a protease with a special Spartan-like protease domain. So we applied this idea to our particular melanogaster system, where we wondered if there's some protein that's cross-linking to 359, and maybe the melanogaster version of MH avoids this DNA protein crosslink, whereas the simulins version sort of aggressively removes these DNA protein crosslinks before the function of that other protein can be performed. So who might that other protein be? Well, in this article from eLife, we found that the substrate of Spartan is topoisomerase 2. And it turns out that imagining that maybe the protein cross-link to 359 could be TOP2 wasn't so far-fetched, because of previous literature showing first that TOP2 actually co-localizes with 359, TOP2 actually cleaves specifically 359, and TOP2 actually is known to resolve entanglements that arise at 359. So the model then is that TOP2 is important for resolving 359 entanglements, preserving genome integrity and fertility. In the ovary, it's important for MHMEL to avoid this particular pathway, while MHSIM might actually interfere with TOP2's ability to resolve those 359 crosslinks, or to resolve the entanglements. So how do we test this model? We test this model by overexpressing TOP2 with the idea that we're then going to override this interference. We do this with the UAS GAL4 system, and here we have our GAL4 only controls where we see a fertility defect. However, when we overexpress TOP2 from melanogaster, what we find is no effect when it's melanogaster in the MH-MEL background. But when we overexpress TOP2 in the MH-SIM background, we see a complete restoration of offspring, just like we saw when we removed 359. And these ovaries have no DNA damage. So these data suggest a sort of second elaboration of this model. Well, we initially thought of this as a two-component sort of system where we have MH co-evolving with 359. Instead, we imagine that TOP2 is actually between these two components, such that TOP2 is evolving to recognize and interact with 359, and MH is actually co-evolving with TOP2. And if this model sort of goes down the road, we would expect that TOP2 would actually evolve adaptively. And we again can conduct our McDonald-Kreitman test. And we did find evidence of adaptive evolution of TOP2. So we think of this now as sort of an adaptive evolutionary cascade, such that the repeat triggers evolution of one protein, which can then trigger the evolution of a second protein. And this is a model that we're actively testing. So I'll just finish with a reviewer comment on one of my grants, which was, how do you know this whole thing isn't very specific to Drosophila melanogaster? Does this have any meaning outside of your specific system? So we've begun to address that, thinking about the idea that satellites are ubiquitous and they diversify across flies, across worms, across mammals, across fungi, etc. And so it's possible that MH doesn't just evolve adaptively between these two species, but instead more generally evolves adaptively across Drosophila. And furthermore, MH is part of a family of genes that all have this Spartan protease domain. GCNA, 2694, 11322 are other genes in melanogaster that also are thought to resolve these DNA protein crosslinks. So we've done some evolutionary analysis on different clades of flies and have found evidence of adaptive evolution across the phylogeny. We see a lot of evidence of gene family diversification, births and deaths of genes entirely, as well as evidence of germline expression evolution from the testis to the ovaries and back again, suggesting that maybe DNA protein crosslink repair is actually chasing DNA satellites over and over again, not just in flies, but in other systems. So with that, I'll just briefly thank my lab and highlight specifically Kara Brand, who is a postdoc in the lab who spearheaded all of this work on DNA protein crosslink repair. And thanks for your attention and your patience with the technical difficulties. If I have a few moments, I'm happy to take questions.
Thanks, Mia. That was really fun. We will stay a little longer for questions if people are willing. Okay, you already have a question. So I'll start with the question from Gary Carpin, but if you are around for a few more minutes, please feel free to type in your questions in the Q&A or raise your hand. So Gary says, great talk and story, Mia. I'm wondering if the top 2 359 interaction is also indirect. That is there an adapter protein that is co-evolving and might be less constrained than evolution of top 2?
I think that's a really interesting question. And I think before we did that McDonald-Kreitman test, we would have assumed, given how important top 2 is and given how much everyone uses it from, flies to humans, that it wouldn't actually be evolving adaptively. The fact that it is evolving adaptively suggests that it does have some flexibility. The other piece of data that occurs to me is just this older paper showing that there's a cleavage site specifically for 359 at top 2, suggesting that maybe it is truly a direct interaction. We certainly need to do a lot more to test that directly. If you had, Gary, any ideas for who that protein might be, I would be delighted to hear about that, to add yet another protein to this adaptive cascade.
Great. Felix Mila Planets has his hand raised. I'm gonna give them permission to talk. Felix?
Yes, hi. That was really cool. I was wondering whether MH and topo2, wouldn't you expect them to be speciation factors? And isn't that inherently something that is something positive? I mean, you spun the entire story as selfish genes and they need to be repressed, but speciation is something I think very important and useful.
Yeah, thank you for that question. You're not the first person to raise that. You're absolutely right in the sense that the speciation community certainly calls any gene that would show what we would call F2 incompatibility a speciation factor. It's very much an incompatibility factor. It is certainly a barrier between these two species. The question of what you call a speciation gene comes up, right? So this is certainly not a gene that is involved in F1 incompatibilities, in the sense that if you make a melanogaster simulans hybrid, we don't think that MH is actually the responsible, factor for ensuring that those species can't, make viable progeny. However, you're absolutely right that any gene that shows these incompatibilities can certainly be called a Dobzhansky-Muller incompatibility, to use, the business speak of the speciation world. And one could certainly enter this question using that framework as well.
All right, thank you. Very cool.
Thank you.
Okay, I don't see any more questions. I will say that Gary said he does not have any ideas for what that intermediate might be. I was going to ask a question, since if we have a little bit of time, but is there a specific part of MH that's evolving? So is it the Spartan domain? And could that give you clues about what is co-evolving with it? Maybe you mentioned this and I missed it.
I didn't mention it, and that's a really great question. So Kara has done a sliding window DNDS along MH, and what she finds is that the Spartan domain, the protease, is very conserved, as you'd expect for a functional protease. But there are 2 regions of the sort of more C-terminal part of the protein that seem to be very diverged. And one, I guess I'll just say that these 2 regions map to the C-terminal region, where not in melanogaster, but in other systems, this region is associated with the sort of self-regulation of Spartan. So it's able to actually cleave itself. And so these are regions of the protein that are involved in that auto cleavage. And so it's possible that it's something that, Kara has hypothesized, is that auto cleavage itself could be sort of the knob that evolution is turning to sort of determine, how much MH is tolerated before it triggers itself to actually start sort of auto cleaving. And so it's not sort of overexpressed and ultimately promiscuously chewing up crosslinks that should be sort of sticking around for that protein to do its transaction. So that's one possible mechanism, is this auto cleavage.
So would you then speculate, or maybe this is something Kara's done, if you just swap out the C-terminal tails, would that lead to the same defect? Is that something that...
That's certainly on the list of items to do. We're very interested in that, and making chimeras is a really natural next step. Thank you for that.
Okay, I don't see any more questions. So since we are at 5 past the hour, thank you everyone for joining. We'll see you again in a couple of weeks. We have a mini symposium planned. And thank you to Mia and Pia for the wonderful talks today.